the main unit of survival really in animals and things in the wild is populations. Now, populations is kind of a very broad term, and if we're going to be able to conserve, manage, or protect things, species in the wild, we need to know how those populations are changing over time. And they're very simple questions to ask, but they're actually really difficult to answer. Things like, how many are there, is actually really tough. You think, well, you just count them, but then you imagine trying to count every crab on a beach. It gets very hard very quickly. Also, it sounds great when you say, well, there's 1 million crabs, but what if they're all males, or what if they're all females? That's not a very healthy population. So not only do we need to know how many there are, but we also need to know more about that population's demographics, their age structure, the sex structure, how things change seasonally over time. Are they there the whole year? Are they only there in summer or winter? All these sorts of things all come into play, and we need to try and find the answers to these if we're going to know if our populations over time are increasing, decreasing, staying stable, or if they've got any major changes that are changing what was happening previously. So one of the best methods that I use for sharks myself, this is the same problem we have. How many are there? What's their population demographics? I use what's called a baited remote underwater video, or a BRUV for short. Very simple device. Down in here you've got bait that sits in the bottom, and bait will attract sharks and fish and things to underneath this cap here. Inside there's a camera, and the camera points downwards, forming underneath. So as a shark comes through to investigate and find out the delicious smelling bait, using those sensors that we learned about earlier, using that chemosensory smell, comes through, and we can get a look at its species. We know how big this BRUV is, so we can estimate its size. Sharks are very easy to tell whether they're males or females, so we can figure out the sex ratio. And then we just do that a bunch of times in different locations and at different times of the year to figure out seasonality and location. So it's a very simple method. Going back, you may remember that in topic 3, level 1, if you want the specific video, it's GTV 3.8. Tamlin talked to us a little bit about using photographic imagery of whale tails to figure out which individual is which specifically. Now, using a baited remote underwater video, we can actually do the same thing with certain species of sharks. Certain sharks have got patterns on the tops of their bodies which are unique to those individuals, and so we can use those to figure out exactly who is who. Using that, we can then put it into a larger method which we'd like to call Recapture. Let me take you to the beach and I'll show you how this works in action. It's gone down a bit. Recapture basically works on the idea that if we capture a proportion of the total population, mark them in some sort of way that we can recognize, release them to mix back into the population, and then recapture a second sample, we can use the ratio of how many animals we've seen before, thanks to the marking, and how many are new to figure out an estimate of how large that population is. So before I said, how will we figure out how many crabs there are on a beach? Very hard to do, but using this method, you only need to capture a small subset of them to figure out the larger population. If I have to lift this guy up, we might find a crab. There we go. So this is our common shore crab. What we could do is mark this guy on his shell with a little white dot, and then we would release him back into the population. We'd come back later, we'd try and find the same crab again, or any other crabs that we'd marked, along with all the new crabs that we hadn't seen before. So this method I've just described on the beach of Recapture is formally known as the Lincoln-Peterson-Chapman, and it's a clever little formula that was come up originally to try and estimate how many ducks were available for shooting seasons in America. But it actually works on wild populations of all kinds. So here I've given you the formula and how it works, and we've got a little example to show you how this would work if you were to do it yourself at home. So the formula works that your population, which is N, equals the number of individuals you find the first time you go out and catch things to mark, multiplied by the number of individuals you capture the second time you go out to look for those marked individuals, divided by the number of individuals you found that had marks on them. We add +1 to each one of these so that you can never end up with 0. So the formula always works. So if you ended up with a 0 down here, the formula wouldn't fall apart. And to balance it out, we -1 right at the end. So to give you a quick working estimate, if you had to go out and measure snails, for instance, you found the first time you went out 236 snails. The second time you went out, you found 193 snails, and in that 193 snails, 40 of them were the marked ones that you found previously. Now, we can suggest possibly a day sort of time between going out your first time and your second time, just to give that population a bit of time to mix back through. But in our example, the top line here equals about 45 or 46,000 individuals. We divide that by our marked individuals as a part of our population, and in the end, the estimate we end up with is about 1,120. Now, this is a bit arbitrary. It just shows you kind of how the formula works. You yourself will need to go out, you'll need to designate the space you want to test in, and you also need to decide on how long you want to look for. So however long you look the first time is about the same amount of time you want to spend when you look the second time. And that way it just keeps everything reasonably equal, because one of the big assumptions that this needs to work is that all individuals have equal chance of being captured. And so if you spend a whole bunch of time on one of the extra capture sessions, that unbalances that a little bit. There are some other important things to consider when using this method. Like I said previously, this is for a closed population, so we can't have any additions or deletions, births, deaths, immigration or emigration from the area. And we're also going to assume that any marks that you put on stayed on those animals, and that you were able to actually identify those marks when you saw them again. You didn't misidentify another white spot on the snail, or that those white spots didn't rub off overnight or something like that. So there is quite a few little assumptions you've got to make to it. Most of the time though, these kind of come out in the wash, and the estimate you get at the end is usually quite reliable. So Recapture can be used on basically anything, and it is often used on larger animals, like my sharks that I mentioned before, as well as the whales that Tamlin told you about. I'm going to hand you back to Hannah, who's going to tell you a little bit more about using these kinds of methods on bigger animals like that.
Hi everyone. So some of the animals that I want to tell you guys about is birds and mammals that you find in the coastal areas of New Zealand. So two species that are really interesting and that has methods of tagging that we've had for decades now is the New Zealand sea lion and the yellow-eyed penguin. So for the sea lions, there's been an ongoing project where sea lions get tags on their flippers. So for us humans, it would be like getting a piercing down here, and that little tag or piercing will then have a specific number for that individual sea lion. That's a kind of tag that we can then look at and try to figure out where that specific sea lion goes, who it hangs out with, how big the population is as well in those areas. For Hoiho, we have been for a long time used microchipping, and we've also started to do GPS tracking on those. For you guys to get involved in surveying birds and seabirds around New Zealand, there's a few ways that you can do it. So first and foremost, if you have a pair of binoculars, they're really good. There's a few markings that is already existing on birds. For example, albatrosses will have some bands on their legs, and they will have specific numbers. So if you ever come across one close enough with your binos to read it, you can report that. The same with the sea lions. If you see a sea lion on the beach and you can read the tag using a pair of binoculars so you don't get too close, then you can report that as well to either the Sea Lion Trust or to DOC. So this is a young sea lion that we have that is tagged. So it's got a tag number 3169. And I know that because I've gone a bit close to it, still kept my distance, a very safe distance of around 10 meters, and then used my binoculars to read the tag. So this is something that you guys can do as well. If you come across a sea lion and you brought your binoculars, or maybe you brought a camera with a good zoom, you can then have a look and see what the tag number is. And also, really important, what color the tag number is, because every season we've used different colors to tag sea lions. So by looking at the color, we kind of can figure out what year they're from. Another way that you guys can survey some seabirds around the coastal areas is by doing some strip surveys, so some transect surveys. Basically what that means is that if you want to survey, for example, down this beach, you start a specific area. It could be one end of the rocky seashore. You go down along the beach and you go in the same pace all the way down in the area you want to survey. So that little walking path that you're creating, that's your transect. And once you're going down in the same pace, you then record down in your notebook every kind of bird that you're seeing. And it has to be within kind of the same width of that transect. So you can say 50 meters that way, 50 meters that way, or if you have a pair of binoculars, you might even go up to 100 meters, and you write that down all the way along your transect. So that's a strip transect survey of bird species. Another method that you can use is called a 5-minute bird count. And what that one is that you stand still, and for 5 minutes you record down every kind of bird that you see and you hear. And then you move along to another point and you do 5 minutes there. Every bird that you see, every bird that you hear. So in order for you guys to do it, it's a really good idea, again, to have binoculars and maybe even have a bird identification book with you. On a boat, you're going to work on different conditions than if you're working on a seashore like this. So let's go to the boat and have a look at some methods there. So here we are on the research vessel Beryl Bruin that belongs to marine science at Otago University. And as you guys can probably tell, there's plenty of evidence of presence of seabirds around here. So when you are on a research vessel like this and you want to survey some birds, you can actually both use the strip transect survey and the 5-minute bird count. But when it comes to the strip transect survey, instead of you moving around in the same pace along a transect, it will be the boat moving around, and you will stand still and note down all of the birds that you're seeing while the boat is moving on the transect. You might even benefit from having more than just one person surveying and having maybe a person on this side looking that way and a person on this side looking this way. You will still have to communicate though, because one of the rules when it comes to the strip transect survey is that you don't record a bird twice. So if a bird flies off and moves around, you might just want to let your partner know about that. The other bird survey method that we talked about was the 5-minute bird count. And so for that one, every so often, and it's up to you how long it's going to be in between the 5-minute surveys, but you stand on the boat and you for 5 minutes record every kind of seabird that you see, the number and the species as well. So now we've introduced you guys to some methods that you can use to look at the populations of different animals. And next time we see you, we are going to try and formulate some questions that we can answer by using these methods as well.